Hello everyone, I'm Sona Charles, working as a PhD student in Bharatiya University, Coimbatore, India. Today I'll be presenting a part of my PhD work entitled Transcriptomic Meta-Analysis Reveals Biomarker Pairs and Key Pathways in Tetralogy of FALLOT. Tetralogy of FALLOT is a cyanotic condition that constitutes about 10% of the congenital heart disorders. Several genetic factors as well as non-coding RNAs are known to be involved in tetralogy of FALLOT. Here in this study, we applied two sparse classifiers, Poisson's linear discriminant analysis and womb nearest truncated centroids to select biomarker genes and miRNAs. A Poisson's distribution is a tool that helps to predict the probability of certain events from happening when you know how often the event has occurred. The womb nearest shrunken centroids is a sparse classifier that models the mean variance relationship using the womb method and incorporate womb's precision weights into the nearest shrunken centroids classifier via weighted statistics. So in the study, we applied both these classifiers to select biomarker genes and miRNAs. So this is the overall methodology of the work. The raw sequence data of the genes and miRNAs were downloaded from the SRA database. The data was pre-processed using the standard pipelines, and about 86% of the reads were mapped to the reference genome HG90. Differential expression analysis was also performed on this data. The weighted gene co-expression network analysis package in R was used to identify the correlated genes from the set of differentially expressed genes. The count data was normalized using the D-seq mean ratio and log transform variance stabilizing transformation method. The count values were then divided by the size factors. The normalized and the transformed data generated were divided into the training and the test set in the ratio of 70:30. The models were fitted using womb NSE and transform PLDA using the R package MLSeq. Five-fold cross-validation was done with a tune length of 10 and repeated 10 times for both the classifiers. The gene selection was performed using womb NSE and transform PLDA. The predicted biomarker genes were checked for the presence of binding regions by the predicted miRNAs. The correlation between the expression of miRNA target gene pairs with significance value of 0.523 was selected for further analysis. Since the miRNAs are expressed inversely with respect to the genes, we checked the expression pattern of the extracted pairs of interactions. The gene ontology analysis was performed on the biomarker genes. Now moving on to the results, 2,757 number of genes were found to have log 2 fold change of greater than 1 or less than -1, and out of which 964 were downregulated and 1,793 genes were upregulated. A total number of 214 miRNAs were found to be differentially expressed, wherein 103 miRNAs were upregulated and 111 were downregulated. The weighted gene co-expression network analysis was performed on the differentially expressed genes to identify the highly correlated clusters. We obtained 5 clusters based on KEGG pathways. The turquoise module consisted of the highest number of genes, while the green module consisted of the lowest number. Gene and miRNA selection were performed using the transformed PLDA and womb NSE. The transformed PLDA was able to select 72 genes and 38 miRNAs, and womb NSE selected 2 genes and 5 miRNAs. The validated miRNA target gene pairs from a pool of selected biomarker candidates were extracted from MIRTAR-based database. The pairs were then chosen in a way that only those which had experimentally verified interactions through reporter assay or Western blot were considered. A significance matrix was constructed to display the level of expression correlation between selected gene pairs. 36 pairs were rejected due to being non-significant. The gene ontology terms for the significant genes were extracted, and the thickness of the edge replicates the percentage of overlapping genes, and the size of the nodes indicates the number of genes. It is clearly evident from the biological process that the selected genes are key players in development and cellular differentiation. the cellular components being associated with collagen and extracellular matrix, which indicates its role in remodeling. The mRNA and target gene expression occurs antagonistically as well as synergistically. There are 14 pairwise interactions between the mRNAs and genes, and out of these 14 pairs, 10 were showing opposite expression pattern, which were further used for the pathway analysis. Focal adhesion and PI3K AKT pathways were found to be more enriched with biomarker pairs. So, as a summary, our pathway analysis is consistent with the findings of the study by Walsh et al. 2006, which clearly mentions that the AKT pathway is highly significant in heart development. The conditional downregulation of focal adhesion kinase leads to congenital disorders, especially in tetralogy of FALLOT. Several studies have identified the mRNA gene pairs as key players in diseases. The significance of the study lies in the fact that the selected biomarker pairs have supporting literature evidences as well and may be used for further experimental validation. Thank you everyone for listening to the presentation. Feel free to drop in your queries and suggestions. Thank you so much.